of Technology, Delhi. And today he will be delivering a lecture to the, it is a, I think, a topic. What is the topic, Saurabh?
It is related to the textile industry, dyeing Nexus, a microbial approach for the textile industry.
Dying Nexus, Microbial Consortium, Mediated Bioremediation for the textile sector. Correct? It's a very, you see, this is a very, very innovative research topic. Today in India, many research workers are working on dye degradation. Because I know Dr. Sanjay Govindwar from Kolhapur. He has many research paper if you search on the Google. On the different type, because there are different types of dye. And he is working on dye degradation, that is what we call it as a bioremediation. And whenever we are talking about the bioremediation, consortium plays an important role. And your research is might be definitely interesting, Dr. Saurabh.
Yeah.
All our students, more than 24 participants are attending today. So they will be very happy, and your lecture is being recorded. Prathamesh, have you started recording?
Yes, sir, I've started recording.
Okay. So tomorrow again it will be circulated. Keep in mind. So I appeal to Dr. Saurabh. Please start the lecture. People are waiting for your PhD knowledge. Thank you. Thank you.
Hello. Yes, I'm audible to you.
Yes, you are audible. Clearly audible.
Yes. Right. So my screen is visible to you all?
Yes.
Yes. So, right. So I'm Dr. Saurabh Samuchiwal, currently an assistant professor at Department of Biological Sciences and Engineering, Netaji Subhash University of Technology. So I have completed my PhD from IIT Delhi in 2023. So currently I'm thankful to the Microbial Society of India for inviting me to present my research to in a PhD lecture series. So my research topic is the dye Nexus, a microbial consortium-mediated bioremediation for a textile sector. Yes. As we know that the textile industry play an important role in India. Around 2% of GDP is contributed by the textile sector. However, dye are currently used in the textile sectors, which is in very high amount. And they result in the discharge of dye effluent to the river water bodies. Currently, we can say the Rajasthan, as we know that the Rankila Rajasthan. There are several textile clusters present in Rajasthan, but they paid a huge cost for this thing. The effluent discharge from this textile clusters result in the pollution. These are the different type of cluster which is present in India related to the textile sector. Some of them are the cotton, wool, and silk. This is one of the river situated in the Rajasthan near the Pali district, Pandiri, and its lands are dying due to the dye. Basically, the color effluent which is generated from a textile sector surrounding this river contributed in the high pollution to the ecosystem. There's a one farmer who said that when this land will grow anything of worth again. Means the land nearby this region is totally deteriorated. They lost their productivity yield. So what approaches? Right now, we are come to the topic that how can we make a sustainable technology for the treatment of this type of textile? So basically, what is the approach? So when we start with the textile sector, so firstly, we have to understand that what are the basic characterization of a textile effluent? What are the physiochemical parameters? After the coming to the knowledge, we can identify the dye group present in the textile effluent, or we can see the wastewater. Then we have to do a particular study which is related to a dye degradation. For example, we have to take only the one dye and then we have to degrade that. Then there is a second process, the optimization of process, in which we are using the temperature, pH, and HRT. And finally, the textile dye effluent treatment, basic on the basis of the proof of concept that we have achieved during the optimization process for a dye degradation. We have used that process for a textile effluent treatment, then the pilot scale study, because pilot scale study is one of the important parameter that we need to study. Otherwise, the industrial are not able to adopt our technology. So this is a long-term performance. Then the market feasibility. How can we decide what is our market feasibility of our developed technology? So we have to do the economics, techno-economic analysis. Then we have to do the life cycle assessment. Life cycle assessment is the tool to detect the carbon footprint which is present or associated with our developed technology. So these are the whole step which we have to do during the development of a new technique for a textile wastewater treatment. So they are the several decentralized treatment approach, that is a physical, chemical, and biological. As we know that we are currently in the microbial and enzymatic phase because they are the sustainable and as compared to the chemical and physical method. So this research start with the specialized microbes. We have to develop the microbes which are specialized for the bioremediation, for the degradation of dyes. Then we have to use that microbes for a degradation. So means we have to validate the process at a TRL level of 4 to 5. After achieving the validation of process at a lab scale bioreactor and a pilot scale, we need to conserve the microorganism. Because when we are using a microorganism and when we are doing with the technology transfer, there is a high chance of contamination of the microbial strains. So to reduce that or to reduce the risk of that thing, we need to make a storable product which can be transferred from one place to another places. It's increased the market feasibility of our microbial consortium, which is developed during the process. So challenges for proper handling of textile. There are the several challenges which are present in textile effluent and need to be rectified. For example, collecting effluent, they are highly alkaline in nature, hot and high salt concentration. Microbes, due to the presence of highly alkaline nature or hot, they lose their viability under the stress condition. Microbial growth, because as we know that microbes require carbon sources for their growth, so they need the nutrients. So these are the major challenge which we need to tackle when we are dealing with a textile. So what are the biological methods for the treatment of textile? Basically, they are the basically two approaches. First is the aerobic treatment approaches, another is the anaerobic treatment approaches. And in case of aerobic means the presence of oxygen. Anaerobic means the absence of oxygen. So what are the pros of this process? In aerobic process, they are easy to operate, high COD reduction. Basically, here COD is the chemical oxygen demand and BOD removal. BOD is a biological oxygen demand. And this process is highly stable. But what are the major disadvantages of this process? Huge sludge generation. As we are dealing with the ASP process, that is the active sludge-based process, which is currently used by a different treatment, municipal wastewater treatment, but they lead to the generation of huge volume of sludge. Less color removal because they are not able to degrade the dyes. In case of anaerobic treatment process, they are have a high declaration potential toward the dye degradation. And they also have another plus point that they have a less sludge production, that is 0.5 to 1 kg per kilo. But there are the several disadvantages. Harmful gas generation. regular maintenance, they need to do the regular maintenance of the system or anaerobic chamber, low COD removal and POD removal. So these are the some major challenges which are associated with the biological treatment. So how can we improve that thing? There's a need of improvement in the biological system. So we need to improve the efficiency and density of dye-degrading microbes. If we increase the dye-degrading microbes abundance in a consortium, then it can directly increase the efficiency. Designing of an innovative bioreactor system. Bioreactor is a very crucial thing which need to be understand and have to develop in a such way that it is responsible for a high microbial growth along with the high declaration potential, long-term storage of the microbes, post analysis. So schematic of a various dye-degrading enzyme produced by the bacteria species and their degradation. So there are the several bacterial species which are having an important role in a dye degradation. For example, Pseudomonas, Enterobacter, Bacillus, Staphylococcus, and Klebsiella. So they are responsible for production of a different type of enzyme, like peroxidase, azoreductase, FNM-dependent NADPH azoreductase, and lactase, well, so there are several enzymes which are present in aerobic stage or under the oxidation stage. And there are the several other enzymes which are present in the reductive phase. So when we are dealing with the aerobic process, these are the enzymes lactase. For example, lactase is playing a crucial role for the tide regulation. But when we are dealing with the reductive phase, when we are dealing with the anaerobic process, azoreductase, NADPH reductase, these are the enzymes which are playing an important role for the degradation. So this is the example of reactive to dye 13, and the upper part show the degradation during the oxidation phase, and lower part showing the bioremediation during the reductive phase. So in my PhD, visit to industrial plant and IO, because initially when we are reading a paper, we are generally dealing with the single dye or dye issue. But when we are dealing with the effluent, so there is a very high concentration of dye, that is 10 to 15 gram per liter. So that is very high as compared to the dye which is used in our synthetic effluent. And they have the complex dye. Along with the dye, they are using auxiliaries, chemicals, salts, and a number of things which make the process very difficult for the microbes to degrade. So these are the things. So this is the process flow diagram. So when we visit the industry, so we have see the process in the textile sector. So there is a pre-treatment range. So when we are dealing with the textile sector, so initially there are the several desizing, scoring, or bleaching unit. These are present in a pre-treatment range of bed operation in the textile sector. Desizing means the removal of the starch from the fabric. Scoring is responsible for removal of the impurities. Bleaching is imparting the whitening to the fabric. So these are the process wet operations which are present in the textiles industry. This lead to a generation of effluent, that is a PTR effluent, pre-treatment rich effluent, that is 0.7 to 0.1 ml. And after that processing of this fabric, there is a musculation phase, then dyeing phase. This is the phase where the dye is used by the industry to impart the color to the fabric, and finally the finishing phase. So this is a one type of effluent, that is a PTR effluent, and this is the second type of effluent, which is highly colored mixtures. So we are targeting the two effluent, is the PTR and dye, and overall it's in a final effluent streams, they combine in a collection type. So the first point, this is the first point where the dye, where the color effluent is get discharged into the final stream, waste stream. So currently the industry uses the ESP process, that is an active sludge-based process. So the primary, it can include the primary statement, that is a neutralization, cooling, and green. Neutralization here required to neutralize the alkaline nature of an effluent. Cooling is to making the cool down of the effluent, because initially the temperature of the effluent is very high. So then there's a biological treatment process. Here the active discharge-based process basically they are using, and sedimentation process. In sedimentation process, the industry discharge result in the generation of huge toxic sludge, because the biological system they are using are able to absorb the dye on the surface, but not the degradation. So they result in the generation of a toxic sludge that is 18 to 20 ton per day. And finally, they are using the reverse osmosis treatment process to remove the color, and this also generate a high ME salt. ME salt here means the multi-evaporator salt, and it is around 15 to 16 ton per day. So these are the basic characterization of a different effluent, that is the desizing, scoring, bleaching, and etc. So here the PTR effluent here contribute high COD and BUD. So the high COD and BUD is due to the presence of starch, enzyme, chemical, etc. In case of dyeing effluent, which is coming from the dyeing unit, contain the high color. But here we can see the COD and BOD is not so high. So in our mind, it's come to that here why there is a high in BOD and COD in the PTR. It is due to the presence of starch. So our microbes can use this starch as a nutrient source. So we can use the PTR effluent as a nutrient source for a microbial growth, and that microbes can degrade the dyes present in a dye effluent. So we can. This is a very cheap sources of nutrient for our microbial consumption. This is the amisol, which is generated by the textile sector. It is highly color and toxic, but we can see one thing here, it contains high NaCl. So the high NaCl content here means there is a chance to extract the NaCl from this amisol, and which result in making a product, byproduct, useful byproduct. So problem associated with the text convention process, as we can see, this is the convention process. ETP, inlet of industry, then there is the aerobic stage, RO, ME unit, and then activity search processes. So here we can see the other, this other different parameter, that is a COD, high COD present in the ETP. If inlet, the color hazel is very high. After the aerobic stage, the outlet have a color, not so much color, but high color, that is 750 to 900 hazen, and low COD, that is around 160. So what are the major problems? Color still remain after the aerobic stage. Our membrane, the color remain in the aerobic outlet, lead to the fouling of our membranes, which is the second stage, and it is very difficult to treat. Third is the ME salt, which is coming, doubling from the RO per minute, that causes the 50 to 60 ton of ME salt generation per day. So it is very highly colored. So these are the things that we need to understand, and we have to make our process or the technology that are able to target these challenges. So my research start with the development of a microbial consortium. We have developed a consortium that from a PTR effluent under an anaerobic condition at 35 degrees Celsius. So we have here filed a joint patent with the application number this with our industrial partner. Then this is the process. Initially, we have tried several carbon sources, that is a glucose, yeast extract as a nutrient sources, and PTR as a carbon sources, because we can see that the PTR contain a high COD and POD content due to the presence of the starch. So these are the different trial that we have done. So our process is able to sustain at a very high range of temperature from 30 to 55 degrees Celsius. Means this process can able to, it can treat the textile effluent at a high temperature, that is 55 degrees Celsius. This process is able to create the dyes from a pH from 7 to 11, means it can work in the alkaline range. So our microbes are acclimatized with this condition. Agitation means the 150 RPM, so they need the mixing properly. Incubation time is around 48 to 72 hours. So we can see here the color removal, there is no significant difference in the color removal efficiency between the 48 to 72 hours. That's why we have selected the 48 hour as a suitable time. So we have tried this our Microbial Consortium for a degradation of different type of dyes. Here we can see at 100 mg per liter, we are able to degrade the dyes using our consortium. So this is the overall diagram of our consortium that for a textile effluent. So here is a PETR effluent we are using, that is in the amount of 30% volume by volume. In case of dyeing, we are using at a 70% volume by volume. After mixing it, we are inoculating with our microbial consortium. Then in the absence of oxygen means anaerobic condition, at a bioreactor at a 5 liter, we are able to degrade our textile inlet, effluent inlet to this level with a pH of 7. So means we are able to achieve 73% degradation in a 48 hour. So this process is consistent at a high temperature, that is 50 degrees Celsius, even at a high pH, that is the alkaline range, 12 pH. Stability of our micro consortium community structure after long-term population. So we have started our research in 2019 and we have ended it in 2022. So the composition of our microbes in our microbes in 2029 year still remains. So means after even a 4 year of gap, the microbial consortium is still able to retain the specific in the microbes which are responsible for dye degradations. Means the abundance of our microbes which are responsible for dye degradation is increased during this interval. So what is the metabolic pathway? So we have to understand what is happening, why our dye is degrading or the color, it become colorless. So to understand this thing, we have taken a reactive group 13 as a sample dye for degradation. Why we have selected the reactive group 13? Because it is majorly used by the industries. Currently, the industry use the azo dyes. More in our India, maximum in the textile sector are using the azo dyes. That's why we have chosen the reactive azo dye. So we have used our anaerobic consortium, and then after do some metabolite analysis, FTR, HPLC, GCMS, we come to know that what is the proposed mechanism. So this is the thing that bacteria translate and different type of enzymes is produced in our process, that is the NADP, quinone reductase, pyridoxine reductase. So this all more like are the azoreductase in families. Some other filtrate alcohol reductase, aldehyde dehydrogenase. These are the enzyme which is produced from the bacterial consortiums. When so this lead to the azo bond cleavage. Azo bond is the N double bond end which is present in a dye structure. So and desulfonation, de-reductive cleavage, and dechlorination. All these processes are responsible for degradation of this dye into a simpler compound. They are mainly the thin compound or amine groups. So this is the pilot scale anaerobic reactor that we have developed at industry. So it have a volume of 200 liter. So this is a schematic of our system that is a smart. We gave the name smart sequential microbial based anaerobic aerobic reactor technology. We have operated at a TRL technological readiness level of 7. It contains the first unit is the anaerobic stage. Then there's a filter. Filter is to use to retain the microbes within the system. Then the aerobic tank, it used for the degradation of residual COD and BOD present in an anaerobic outlet. Then sediment and aqueous stream. So this is the actual image of the plant which we have developed in the industrial sector. So anaerobic stage 200 liter membrane module having a 2 filter system 0.5 to microbial filter. Then there's an aerobic reactor, then activated carbon as a polishing stage. So when we are dealing with the things that the color it have initially in inlet stream very high hazel 3000 and COD 2400 mg per liter present in our textile effluent. After anaerobic stage, that color decolorize, but turbidity still remain in the system. So it's mean the poor solid liquid separation means still the microbe is suspended in the system. After every stage, still pale yellow color is present. To do the ACF process, we are able to remove the yellow tinge present in the thing. So the ACF outlet does not contain any fission or COD. That's mean the ME salt, which is developed from this ACF, using the ACF are able to produce a ME salt having a very light color or means white color. So when we are dealing the meta elemental analysis of this salt, it will contain 95%, more than 95% NaCl. So this is the long-term operation result of our system that it shows the consistent performance. Initially, we have tried our Microbial Consortium to develop a project, but this is the initial state, and we can able to develop a project to decolorize the dye and retain the microbes within a dialysis membrane. So this is the outlet of our developed smart process. So here we can see the inlet color. Inlet, here we are using a textile effluent, then we have a smart process. The aerobic sludge will be used in the cement water, absorb and not breaks. And the salt, ME salt, which is generated from the ACF effluent, can be used for the development of colorless salt using a heat treatment. Then the developed salt is used in a pad batch process. Pad batch process is a process which is present in a textile sector itself. So means it's the inner recycling of a byproduct within the same industry. Why we are using the pad batch process? Because when we are treating the fabric with the high NaCl, the fabric is able to retain the dye on its surface. Means it's increase the dye capture efficiency of a fabric. So this is a comparison of our smart process with the conventional method. As we can see here, the smart process and conventional process. Conventional process here is the aerobic stage, reverse osmosis, and multi-factor evaporator. But in our case, we are able to degrade the color more than 90% and COD. And the sludge which is formed from our process is 3 kg per kL. But in case of conventional, it is 5 to 6 kg per kL. And then the multi-factor evaporator lead to generation of ME salt, and industry have to pay around 66,000 rupees per ton for their disposal. But in our case, we are able to recover around 14 to 15 ton of ME salt, and we can use it for a NaCl demand of our industry. And so it's save around 80,000 rupees per day for the industry. So what is the commercial potential? So commercial potential here we can say that smart process cost around 9 rupees per kl, and ASTs cost around 70 rupees per kl. I'll discuss how we have calculated the 9 rupees per kl in the next slide. AOP is the advanced substrate process, which is very high costing process. That is, it goes 155 and 9 rupees per kl. So it's very costly process, and our process is robust. It can operate at a high pH, high temperature, and one more thing, we have tested our consortium for 7 different textile effluent, which is collected from the different industries situated in the northern India. So why smart? Now there's a need for the modification of the process. As I told you that the turbidity still remain in an aerobic stage in a smart process. There is a high turbidity still present in our anaerobic stage. So what are the problems? So this is due to the suspended cells. So poor settling lead to the high turbidity and continuous washout. Means if the cells is still suspended in a system, then it leads to the continuous washout of the microbes from our anaerobic chambers and directly lead to the decrease in the density of our microbes. That's why we need to retain the microbes within the system. How can we do that thing? So how to prevent the cell wash from the reactor? So we need to identify a suitable membranes or material to retain the microbes within the system. That is the development of a modified reactor. So we need to design a bioreactor having a membrane or some attachment material. Focus on preventing the microbial washout by enabling the cell attachment to the surface. Basically, the material or membrane which we are using are able to retain the microbes on their surface. So this is the one of the critical parameter that we need to identify. And validation of the process by on-site validation at the 200 liter, because the textile pilot scale validation is a very crucial step before understanding its market feasibility. So we have tested two type of things. Membrane technology, we have to make a membrane that are able to retain the microbe within the system, or we can use a microbial attachment. This is the second strategy. Here we can add some attachment material that can able to retain the microbes on the surface. So what are the important characteristics of the microbial material which is required to retain the microbes? So for example, surface roughness, porosity, surface charge, hydrophilicity. These are the different type of parameter that we need to understand. So bacterial attachment to the surface means the surface charge. So if the microbes have rough surface, then there is a high chance of a bacterial attachment. And we know that the bacteria have a negative nature. If the microbials if the membrane have a positive charge, so there is an attraction between the bacteria and the microbes. If our material have a negative charge, which can be determined by using a zeta potential, then there is a repulsion. So it is a lead to the repulsion between the bacteria and the material. And hydrophilicity. So we need to understand what is the hydrophobic nature or hydrophilic nature of the material. So these are the important parameters that we need to understand while designing a reactor. So these are the some few literature that they are using a ceramic membrane module as a membrane to retain the microbes. And here in the second thing, they are using a PF membrane as a carrier for the attachment. So these are the two examples that literature using for the attachment. But in our case, we are designing a smart process. Here we are using microbial hollow fiber membranes in our systems. Why we are using the hollow fiber membrane? It is basically it have a high surface area for the filtration and able to retain the microbe within the system, and they are not. We have also tested the pressure, DMP pressure. There is transmembrane pressure. They can operate at a high transmembrane pressure. So it is the new design of the like smart process, which is present in the textile industry, and we have operated it in the industrial processes as well. So microbial product development. The last thing, how can we develop microbes? So we have developed a microbial by making a capsule. One part contain the nutrient sources, another part contain the dry microbes, which contain the microbes along with some additives. So this capsule is able to treat the textile effluent, and we have tested this capsule for a different textile sector. And when we are coming the cost, it's cost around 8 to 10 rupees of a particular capsule. And one capsule is able to treat 10 liter of effluent, and that effluent can be used as an inoculum for upscaling the process. So means that this capsule is act as an inoculum as well as a seeding material. Post analysis of a smart process. So how can we determine the cost? So cost we are here using operational cost, not operation capex. Not we are not dealing with the capex here. We are dealing with the opex. So what is the requirement of daily power consumption during the effluent treatment? So we have calculated on the basis of aerobic tanks, and it's cost around 6.9 rupees per kg. But when we are dealing with the e-extract, the cost is slightly increased. So one research could be done in the place that we need to replace the e-extract that we are using as an nitrogen source with the other low-cost alternative nutrient sources to make our process less, to decrease the cost of our process. Then we have done the life cycle assessment. Here we are using the SIMAPRO software, and we have developed our technology. So to determine the life cycle assessment, it's basically we are comparing our smart process with the ESD process. Here we have designed a functional unit of 1 KLD. So these are the result of our smart in comparison with the ESD. So in case of smart, we can see here that the smart having a 42% less impact as compared to the activated sludge process. And we have see the impact of our process in 4 different categories, that is a human health, ecosystem, climate change, and resource. In all category, the smart is have a less negative impact to the this category when we are comparing with the ESP process. It means that our process has less carbon footprint, and we are using here an impact 2002 methodology to define the impact. So overall, LC has shows around 41.5% less than negative impact on the environment as compared to the existing process. So this is a roadmap of my research that it starting in 2017 from a textile industry. Then we have to optimize the process. Then we have done some lab skill study in between the 2018 to 2019. In 2019, we have developed some smart process at the tier level of 7. We have understand the major problem. Then 2021, we are able to define the economics and the life cycle assessment. Currently, we are doing some research in the modification phase. So this process or this technology is able to target different SDG goals, that is SDG 6, 13, 15, 12, 14, and 9. So we are able to target a different SDG using our process. So recognition and awards. So my this research got a Best Translation Research Award from a Global Bio India Show 2024. I have received the best thesis in bio mass conversion process, that is the Professor Meera Madan Memorial Excellence Award for this research. Also, I have got the distinction from IIT Delhi, and I have got received the Professor T.K. Ghosh Memorial Award for the best thesis in biochemical engineering and biotechnology from IIT Delhi. Apart from it, I have also received the Environment Award and Young Scientist Award. Currently, I am working at a NSUT, and I have also awarded with the NRF PMCRG project for developing of the photogranule-based advanced photobioreactor for sustainable wastewater treatment, reuse, and recycling. This is from my side. Thank you. Yes. Questions?
Thank you so much, sir. That was such a wonderful presentation. And I hope our students, researchers, everybody must have enjoyed, and then hope they'll get back to you if they have any queries. Even now, if anybody having any queries, please ask questions. We are ready to take up. Sashwati, Rupa, can you please take up the questions from the audience? Any questions from our student and
Ma'am, I have a question.
yes, ma'am.
Yeah, this is Varsha Pegwal. So, sir, have you done any compatibility test just to ensure whether your isolates are compatible with each other?
Yes, we have done the microbial synergetic, and we have tested this thing. Yes, synergy we have done.
Okay, sir. One more question, sir. Talking about that capsule, can you throw some light on that the capsule that you have developed? Like even
Yes.
I'm working on the similar field, not related to dye, but on phosphate solubilizers. So even I'm working on consortium development.
Right.
So yeah, so can you just give me some idea about it?
Yes, when we're dealing with the microbial consortium product development, then we have to use the carriers, because there are the several methods for the drying process. First, you have to understand that there's, for example, electro drying, then there is a lyophilizer, vacuum drying.
Yeah.
You have to use different process for drying, and you have to use also the carriers. Basically, in my case, I have tested for a starch, ascorbic acid, or milk powder or something. So you have to think and you have to design your career and the process, drawing process as per your microbial consortium activity. Yeah.
Okay, sir. Thank you so much.
Any more questions from our audience?
Yes, ma'am, I have a question
Yeah.
to the sir. Just a second. So you've explained about the word called smart, and we being the like we've just entered into our undergraduate into research. So I familiarly am not so aware about the term smart, and also I got this doubt: will this smart be also used? I mean, to other heavy dyes for tannery or paper industries like.
Yes, so we have tested our process that is a smart. Smart is a term that we have given to our technology, developed technology. Yes, we have tested it for the paper industry. And because paper industry equivalent is more than similar to the textile industry, but still in textile sector, there are several heavy metal or another compound which is present. But in paper industry, it contains the high COD. So there, if you are dealing with the paper industry, then you have to mix, acclimatize your microbes with respect to high BOD, right? In textile sector, the BOD level is not too much high, but the color is a major. But our process is able to treat both of them. Yes.
Right. Thank you, sir.
Yeah, any more? Yeah, there is a question.
Chat box.
Yes. Sir, could you please suggest the tool that you have used for preparing the schematic diagrams? Already, this is,
Yes,
yeah, already, yeah, this question is already been.
PowerPoint. I have used PowerPoint only for making a diagram. This all diagram which I used in the PPT is made in PowerPoint.
We also got a question from the
There's no
audience.
special tool for that.
I'm sorry. We got a question from the audience. You've also developed the microbial capsule product. How does it help in field applications and storage compared to liquid consortium?
Yes, when we are dealing with the liquid culture, then we have faced that the microbial consortium change its property when we get transit from one state to another states. And when we are dealing with the microbial consortium, then we have to retain, incubate our microbes within a specific temperature, for example, 4 degrees Celsius or something. But when we are dealing with the so we can capture or we can store our capsule at a room temperature, and we have tested its shelf life, so when we are store it at a 37 degrees Celsius, we can store it for around 9 months, but this is not possible when we are dealing with a liquid culture, right?
Okay. Thank you, sir.
I hope we don't have any more questions here.
Okay.
I'm checking the chat box, sir. Here, nothing is. Anyone from? Yeah, one more is been posted here.
Yes,
Enzyme profiling
I-
of the consortium suggests its application in pharma effluent also. Have you tested any?
no, I have not tested for the pharmaceutical industry. I have tested only for the paper, textile, and leather industry. In case of pharmaceutical industry, we have to deal with the antibiotics and AMR genes. So that's a different research.
Yeah. Thank you so much, sir, for such a enlightening session. Our students have really learned many things from your talk, and it was very interesting thing to listen to your research topic, and also happy to see your achievements. And I
Thank
hope our
you.
students also would follow the same path, because
Oh,
we have many students from a different platform, right from UG student, PG, and till
yeah,
research scholars also, and we
yes,
have faculty members.
Yeah, thank you so much for inviting me. Yeah.
Yeah.
Okay.
So can we request Vasuki to deliver a vote of thanks?
Sorry to interrupt, ma'am. There's a question in the group
Yeah,
chat.
ma'am, there's a question in the group
Okay.
chat.
Yeah, can you please read out?
Okay. Textiles contain substantial amounts of polyester, nylon, polyurethane, elastane, and acrylic fibers. Have you done any work regarding their degradation?
Textile fibers present in the effluent stream that have a polyester nylon, but when we're dealing with the effluent, then we have to treat it. Basically, we have to remove these fibers using a nylon mesh or grid chamber. So when we are dealing with the effluent, then we are not able to treat the polyester because it is not present in a effluent. It already captured during the grid process or a primary sedimentation process, prime grid. Yeah. We are not dealing with the microplastic here. Yeah. Next question. Okay.
Yeah, anyone? I think no more questions posted. So shall I request Rupa to deliver vote of thanks? Vasuki is there.
Yes. Good evening, everyone. As we come to the end of today's PhD lecture series, I feel privileged to propose the vote of thanks. But before, there's one more question in the chat box. Can we go to that?
Yeah. Can we use the biochar as a substitute? Yes. You can use biochar, but when we are dealing with the industry now, it is not feasible. How much biochar you will use for treatment of a effluent? For example, you have designed a process that capture the dye from the effluent using a biochar, but when we are dealing with the MLDs of effluent, then this process is not economical feasible because the biochar have a limit to capture the certain amount of dye present in the system, right? So we need to make a technology that is not saturated with the high concentration of dye. But in case of biochar, it's saturated at a high concentration of dye. And secondly, it is not economical feasible right now. But right now, there are several research currently taken place. So capture the microbes. So yes, it is in the under research. You can use. There's the ball for it. You can use if you are doing the research.
Thank you, sir. First and foremost, I sincerely thank Dr. Saurabh Samochiwal for taking the time to be with us and for sharing your valuable knowledge, research, insights, and experience with us. I extend my heartfelt gratitude to our Microbiologist Society, India, and our institution for organizing this meaningful academic session and providing us with such a valuable learning opportunity. I also thank our respected faculty members, organizers, and all the students and research scholars for their presence and active participation. Once again, thank you, sir, for an enlightening session. Thank you, everyone.
Thank you so much, sir. Thank you all. The goal of research is to turn curiosity into knowledge and knowledge into meaningful impact. Thank you once again, sir, for your valuable time and for inspiring us to become better researchers.
Yeah.
We are truly grateful for this wonderful learning experience. Thank you all.